Welcome Biomode Freshers, we're Team 5, and we're going to start by introducing you to the central dogma of biology, which you may or may not be familiar with. The key concept is the flow of information from DNA to RNA to protein. We know DNA can replicate itself to pass on genetic information, but what you may not know is that RNA can replicate itself in certain circumstances, and indeed it can be reverse transcribed back into DNA. This is particularly helpful for viruses who can reverse transcribe their virus RNA into our human DNA, for example HIV. Crucial to the central dogma is the concept of flow of genetic information, and whilst there are some exceptions to RNA, there are no exceptions with proteins. Once the information has been converted to protein, there is no known instance of it being converted back to RNA or DNA. Why is this important? Well, this is the way in which genetic information acts. It sounds a bit strange, but it's true. And given that all life is governed by this, it is fairly important. The key thing to remember about the central dogma is that proteins are always final.
You already know that every organism has certain genetic information that is stored in the organism's DNA, and you've also learnt in high school that cells and the nuclei are pretty tiny. But you probably haven't learnt how the DNA manages to fit into such a tiny nucleus. After all, humans have enough DNA to reach from here to the sun and back more than 300 times. So how does all that DNA fit into such a tiny nucleus? The answer is packaging. At the simplest level, we think of DNA as a double-stranded helical structure. The process of packaging begins with the assembly of nucleosomes, which are the building blocks of chromatin packaging. These nucleosomes are formed when 8 special units, called histone proteins, attach themselves to the DNA molecule. Histone proteins are positively charged proteins, and since DNA is negatively charged because of the phosphate groups, the histones bind with the DNA very tightly, and this allows the DNA to wrap around the histone protein. This structure resembles beads on a string, where the histone proteins are the beads and the DNA molecule is the string. The beads on a string structure then manages to fold even further to form a 30 nanometer fibre. This happens because the nucleosomes coil together and stack on top of each other. This process doesn't stop there, and the 30 nanometer fibre forms loops that are 300 nanometers in length. The 300 nanometer structure folds and coils even further to produce a 700 nanometer fibre of condensed chromatin loops that's 250 nanometers wide. Further tight coiling produces the end product, which is the familiar structures called chromosomes.
You have all probably learnt about semi-conservative replication before, but in this course we go over it again in greater detail. As you all know, semi-conservative replication is the process by which all DNA in cells is replicated. The parent strands, i.e. the DNA that is being replicated, separates into 2 strands. These act as templates, allowing the formation of new daughter strands that are complementary. The DNA strands bind to each other via complementary base pairing. Therefore, 2 new identical strands are made, each strand having 1 new and 1 old. The complicated part begins when we learn exactly how DNA replication occurs. dNTPs are added to the parent strands to form new daughter strands. The replication process only allows the nucleotides to be added in the 5' to 3' direction. Only the DNA strand that starts from 5' to 3', the leading strand, is continuous. The other strand, the lagging strand, starts from 3' to 5', so a different process is required. Okazaki fragments are formed and are fused together after replication is complete. This process can be quite daunting to try and remember, but by going over the mechanism many times and picturing an animation of how it works and watching many video clips of the process can be really helpful.
Okazaki fragments were discovered by Japanese microbiologist Reiji Okazaki in 1969. They are short, newly synthesized DNA fragments formed on the lagging template strand during DNA replication. Whilst the leading strand can synthesize continuously in a 5' to 3' direction, the lagging strand can only synthesize in bits, forming these fragments. RNA primers are needed at many points along template strand to allow DNA polymerase to add bases to the exposed OH group. An important point to note is that the RNA primers are used to allow specific degradation of RNA rather than DNA by a ribonuclease, meaning that none of the DNA fragments are lost. The Okazaki fragments are then linked together by DNA ligase.
You have previously learned that cells undergo mitosis. This means that practically all cells should have the same DNA, but why, for instance, does a nerve cell have a different shape and function in comparison to a red blood cell? Well, this is all thanks to the difference in gene expression between different cell types. The changes in gene expression are maintained through cell division without any changes in the DNA sequence. The study of these modifications is known as epigenetics. In this subtopic of the central dogma, you are going to explore the mechanisms of how the cells are able to change the expression of their genes. Though there are many ways to alter gene expression, you will only learn 3 of them, which are DNA methylation, histone modification, and microRNA. As their name suggests, DNA methylation is the process of adding methyl groups to a certain region of the DNA, while histone modifications is achieved by modifying the histone tails. Out of these 3 mechanisms, microRNA-mediated gene regulation is the most intricate. The mechanism is complex not because the concept is hard to grasp, but because you would need to be familiar with a lot of new terms and names, so we won't cover it now. The best way to learn this is by using diagrams such as flowcharts to visualize the aspects of this mechanism you may find difficult.